Our strategy is designed to reduce global warming and air pollution by genetically modifying species of trees to carry out photosynthesis faster, reducing the amount of CO2 in the atmosphere. The most harmful greenhouse gas is carbon dioxide. Although it is not the most damaging per molecule, there is so much of it, and it remains in the atmosphere for so long that it is estimated to cause 60% of global warming. The greenhouse effect occurs as more sun rays than normal are re-radiated back to the surface of the Earth after being absorbed by greenhouse gas molecules. These rays would normally just reflect off the Earth and leave the atmosphere. This causes the Earth's environment to get hotter and hotter due to the extra radiation. The more greenhouse gases there are, the more re-radiation occurs and the hotter Earth gets, causing global warming. These effects can be devastating, including increased rates of extinction, sea level rise, and disturbance of climate patterns, and therefore reduced crop yield. By reducing the amount of CO2 in the atmosphere, these effects can be reduced. Photosynthesis is the process that many organisms depend on to create glucose and oxygen. It has two stages, light-dependent and light-independent. The light-dependent stage causes the formation of oxygen, and the remaining products of the first stage are used in the light-independent stage to form glucose. In the stroma of the chloroplast, carbon dioxide combines with ribulose bisphosphate using the ribisco enzyme to create two molecules of glycerate 3-phosphate. Then ATP and NADP from the light-dependent stage reduce the glycerate 3-phosphate to form TP. This TP can be used to create glucose molecules needed for cellular respiration. Ribisco, officially called ribulose bisphosphate carboxylase oxygenase, is the enzyme that catalyzes the carboxylation of ribulose bisphosphate. It is a very slow-working enzyme, catalyzing only 2 to 3 reactions per second per molecule, and is therefore a severely rate-limiting factor for photosynthesis. If this enzyme worked faster, then it would allow more carbon dioxide to be taken in and oxygen released from the plant. Our idea is to edit the part of the genome that codes for this enzyme to make the enzyme more efficient. By comparing different forms of ribisco that appear in the natural world, we can analyse which are the fastest working and why, then enhance these differences so that they are even faster. The CRISPR-Cas9 system allows us to modify the genome efficiently and accurately. It evolved naturally as a defence mechanism for bacteria against bacteriophages, but has recently been recognised as being able to be manipulated. Scientists can now use it to edit the part of the genome that they want. An RNA guide molecule is designed by the user to match the DNA sequence where the target gene is found. Cas9 is attached to the RNA guide, and the Cas9 scans the genome for a match of the guide RNA. When it finds it, the Cas9 attaches to that section, cuts both strands of the double helix so that the new, altered version of the gene can be inserted in place of the old one. In our case, the improved ribisco enzyme. The most effective growing environments in the world are tropical rainforests. The altered gene can be inserted into a native tree species and then planted in vast areas that have undergone deforestation. This would also create jobs for the local people in planting the trees, which might reduce non-sustainable exploitation of the local area. These trees would need nutritious soil, so workers could also be employed to regularly prune the trees, leaving the branches on the forest floor to decompose and the nutrients reused. Over time, new species of modified flora could be introduced to diversify the area and create a more varied habitat so that more animals could live there. This method would also be cheap, as CRISPR costs very little.